Thank you so much for the kind introduction and also invitation here. I'm really excited to be here for a few reasons. I think one is in May I moved from Boston to Cologne, and one personal item on my wish list was to collaborate more with the BigBrain team and also Katrin's team in general. Essentially make use of anatomy in a clinical setting, and that's what this talk will be about. And then the other point is I'm also super I would say I'm a fanboy, have been a fanboy of the M&I space forever, since I can imagine. So huge fan of Alan's work as well. So being here is a great honor to me. And I will start with the end of my talk, because I think the aim is to convince you that atlases are still cool. And I think you all think so anyways, but hearing this from somebody that uses these things in clinical applications may still empower your work even more and maybe even motivate why you're doing what you're doing. So, because the big problem is resolution in what we're doing. When people invented the MRI, this was fantastic, great breakthrough, but it also, in my opinion, led to the point that surgeons would think we now see everything, we don't need the anatomists anymore, and they were kicked out of the operation room. And that was a big mistake in my view. Still is, and I mean, this was of course a gradual thing, and not even a conscious decision. But I think I still have many colleagues in my field that tell me, "Well, we see everything; we don't need atlases." Right? Now, what they see is on the right, as you can see, not much. It's a bit inflated. This is a vervet monkey brain from Marcus Axel, so it's smaller than the human would be, but it's still like the big brain is reality, right? Is on the top that you can see. These are the cells that we want to see in theory, want to modulate, want to stimulate and read out and everything. But what we actually see in optimal MRI is really not much, as you all know. So the solution to this is you. DBS needs you, deep brain stimulation needs you, stereotactic surgery needs you, so continue what you're doing. And you might see that Uncle Sam has even an AI-generated something on his head. So this is really true, right? But let's rewind. I started with the end. This will be more on this in the end. But I think to make this point, I have to essentially tell you a bit more of the details of my story and our research, which I would say is just partly related to BigBrain, but I think important to know a bit about, to know what we're doing and why we need you. So where do we start? Neuromodulation. Neuromodulation, as you know, is our way to interact with the brain if we don't use words or sounds or music, but we poke into the brain with something from the back door, right? We can do this invasively. We can insert electrodes, but we can also use non-invasive forms, focused ultrasound. We can use transcranial magnetic stimulation and so on, right? So we can classify this as non-invasive forms and like TMS here, or invasive forms. And I mainly talk about the invasive forms and specifically deep brain stimulation as the most, dominant proponent there. But I think everything I'm going to say is applicable to any form of neuromodulation in a way. Some of you may not have heard of deep brain stimulation. Most will, but that's totally fine. I hadn't even heard about this in medical school, really. But it's a very established thing. So it's not high-tech, it's not Frankenstein, and it's also not science fiction. It's been around for 30 years. Health insurance pays for it. It's CE marked and FDA approved and so on, especially for Parkinson's disease. But it's still interesting that not many people know about it. So I will tell a little bit about it as an introduction. You can see it's a surgical technique on the left. This is actually here at Charité. You can see the colleagues there operating, and we have our software also at the time there to assist to some degree. And this is an invasive procedure where electrodes are being implanted into the depth nuclei of the brain, and they stay there put, right? So it's not a one-time thing. You stimulate and go home. You put it in, you put in a pacemaker, you close everything, and it stays there for 20 years if the patient survives that long, right? So, or even longer. So essentially, this stays until the end of life. And then we can sometimes recharge or replace the battery. We can reprogram the battery. You can see This is not a detail, it's kind of important to the talk that these electrodes have multiple contacts, so we can afterwards, once the electrode is in there, still fine-tune it and switch on specific contacts and so on. And I think the best way to demonstrate how effective DBS is typically with showing a video. This is one gentleman I treated here myself at Charité as a resident. And the only difference between the two videos is really that on one, DBS is on one it's off. There's no dopaminergic medication. This is a gentleman with Parkinson's disease. And you can see that the movements are much, much more fluid on the right side where the DBS is on versus on the left side where there's a lot of bradykinesia, stiffness, rigidity, and so on, right? So marked difference. It's not like TMS where you have to use statistics to see something. No, you switch it on, tremor stops, right? It's really that salient. But in deep brain stimulation, millimeters matter. And you can see this here in 3 electrodes that have been superimposed into MNI space. And the active contacts are marked in red, and you can also see this optimal spot that we think is the best target site, and the world concludes this is true, in this small nucleus called subthalamic nucleus. The nucleus is just a centimeter long. So you can see in patient number 21, the electrode is maybe a millimeter off target. And that patient had 80% improvement. You can see it on the scatter plots. On the left, you see, the y-axis is empirical improvement. And then the next one, 51, is just 3 millimeters apart and only had 30% improvement. And then you have one that's really outside of the SDN that did not improve at all. And probably even get worse because we take the scores a year later and Parkinson progresses. So just a few millimeters make it or break it, right? So that's why precision is really key. You can also visualize this differently by looking at recordings from these sites. And just by looking, not as a non-expert, looking at these patterns, you see marked differences that there are neuronal populations that are very different from each other, just millimeters apart from each other in these regions, right? So it really matters where we are for the effect, for what we stimulate, and so on. But then when we do, it really works, okay? And then I think I don't have to convince any of you really that what really matters to stimulate is the brain's circuits, the circuitry. The brain is a network, so we need to modulate not a specific site, but a brain network. And that will, once we know this brain circuit that's producing tremor, let's say, we can now identify where we put electrodes. Could even be 2 electrodes targeting the same network in different nodes of the network, or 3, right? But knowing that the circuit, that's the actual sweet spot or model. And the same applies for non-invasive stimulation as well. But the problem is, how do we define these circuits in the first place? How do we know where tremor lives in the brain, right? It's not an easy question, and we've been trying, the field has been trying for 30 years without real success. I would like, well, for tremor, we kind of know, but for other symptoms, we don't. So, of course, one option is animal models, the classical idea, we start from the mouse, we learn it there, we cure Alzheimer's there, we then translate to the human, just hasn't worked, right? Hasn't worked in Alzheimer's, but also even more so not in mental health disorders like depression or OCD, because these things are different in a mouse than a human. That doesn't mean mouse research is not really important. I'm a big fan of it, but it's just not a good model to come to brain therapies, at least so far. The other option that people have done is functional imaging. So we've heard Kamil speak, fantastic new developments. We could scan Parkinson's cohort and a healthy cohort, contrast them, see what's the difference, and they say that's where we have to put an electrode. But I think nobody in this room would actually do this, right? To actually really use that information and stick an electrode into somebody's head. Because that doesn't work either. It hasn't worked for 20 years. fMRI has not translated to any really clear insights in medicine, therapies. Insights, sorry, it's wrong, but therapies in medicine. So one reason for this could be that the difference that we see are not causally linked to the symptoms, but they are correlated somehow, right? And that could mean that they even compensate for the symptoms, right? It could be network differences that try to compensate for tremor. We shouldn't, lesion or downregulate those. Could also be they are causally linked, or they could just be spuriously correlated. So it's just there's a causality gap there. And the same applies to electrophysiology too. So I don't propose we have the answer, but what we try to mitigate this problem is to reverse it and essentially start with the DBS sites and the TMS sites to come and hone down, triangulate the brain circuits that we need to modulate, right? So we try to see where does it work, where are these working things connected to the same network, and maybe that's our circuit. That's the idea. And as a third modality, we use brain lesions as well, and brain lesions are also causal. You have a stroke, you get aphasia, next day, that's a causal link. It's as causal as it gets in the human brain. Goes back to, Broca and sorts, right? Lesions have been important for brain discovery for a long, long time before us. This is really important. There's not much else in the human brain where we have causal evidence than these three things: brain stimulation sites and lesions, right? So with this in mind, what we do is we try to reconstruct these electrodes with software we created. LeadTBS has been mentioned maybe once or so in this symposium as well, where we try to be as precise as possible and also make this transferable across patients, right? So we can have the electrode localization in patient A and B and C and compare them to one another. And this, of course, involves the MNI space or normative spaces to co-register patients as well. But we really care about millimeters here, right? So we've had to, maybe in comparison to an fMRI study, we had to really reinvent some wheels to make this as precise as possible in that specific region down there subcortically. And then what we can do, let's say for structural networks, we can use a set of streamlines, and we can take, say these are all candidate tracts that could be our circuit, right? And we're making them all white. We don't care about, their anatomy. So we just say that's the candidates. And then we take the computer and go one by one, pick out one of these tracts and overlay our electrodes with the tracts and test for each of them. And this is typically 200 or 500 electrodes across patients to just test who did stimulate this tract and who did not, right? And then there are three classical scenarios. One would be all the top responding patients did stimulate the tract. Then it's a good tract. We give it a positive weight and positive color. And then the poor responders did not stimulate the tract. So that tells us even more. This seems to be a critical tract. The opposite can be true too, that all the poor responding patients did stimulate the tract and the top responding did not. So in that case, of course, we give it a negative weight and we think this is maybe even a detrimental tract. We should stay away from it. And then, of course, anything in between can be true where the tract just doesn't differentiate between top and poor responders, let's say in tremor, right? So we say this is an irrelevant tract, we filter it out, and that's why we call this fiber filtering. So if we do this now iteratively across the whole connectome or these candidate tracts, we see a magic pattern appear of this red bundle that plays a key role to mitigating tremor. And we had electrodes here in the thalamus. This is the cerebellothalamic bundle, and we kind of know this really plays a role in tremor, right? So we have a statistical handle now that modulating this tract and these streamlines in that tract would play a role in tremor. This is kind of the story here, right? What I want to say now to make it all more complicated is that we can also lesion here, and we have the same effect, right? So it's called deep brain stimulation, but we all don't think it's really stimulating. We're actually lesioning or downregulating circuits that seem to be malfunctioning, right? We know this. We can really use focused ultrasound surgery or classical ablational surgery to burn a hole here, and we stop tremor. It works. We can also put in electrodes, and we stop tremor. It also works. And the same is true for almost any of the historical targets where people have either carried out a lesion or put in DBS, right? So there seems to be a downregulation or information stop mediated by a DBS. And this to me means that likely this red circuit here that you see is not the one that we have to stimulate to improve tremor, but that is dysfunctional, that we have to downregulate to improve tremor, right? So you could even think of it as a sort of tumor, right, of a circuit that's become out of check, is probably overactive or dysfunctional, doesn't transmit the information it should be. We are not yet there to fix it, right? To essentially, make a real prosthesis to kind of replace the circuit with something computationally effective. But what we can do is downregulate its activity so that the rest of the brain can, go ahead and do its thing. And so this whole idea became pretty interesting to me when I entered the field of DBS, when I thought, okay, there is this small structure that I have briefly introduced before. It's the subthalamic nucleus. It's below the thalamus. I think many of you know it. You're the anatomists here. But P-down is around 1 centimeter long, and lots of frontal projections project to the STN. And I heard this is the predominant DBS target. This is where most of the DBS electrodes in the world will be placed, and especially for Parkinson's disease, PD here. But I also learned after a few years entering the field, this also works for dystonia, right? And then some other people told me, oh, by the way, it also works for Tourette's disease. And then some other people also told me, oh, it works for OCD even, right? Very different disorders. How can that be? First thought, it's all placebo. This doesn't do anything. You can't put electrodes into the same thing, small structure in the brain, and treat all these very different things. You could even list addiction here too, right? Because there's not as much evidence, but in some individual patients, STN-DBS for addiction also worked, right? So how can that be, right? We did investigate this with a large-scale multi-site collaboration where we, asked all our dear friends across the globe, because every team has their expertise in something different, right? It's rare that one center would treat all 4 disorders and have, good cohorts there. But for example, Tourette syndrome, which is the newest kid on the block for STN-DBS, has only been done really in Shanghai and Pisa, so we had to reach out to these folks. And then, of course, OCD is also not that predominant yet, so specific centers have their expertise there. So we try to pool everything together and investigate this. Massive effort led by Barbara Holunda and Ning-Fei Li in the lab, where they localized all these electrodes. It's painstaking work because it really matters where, the millimeters matter, so it's a lot of manual work as well. If we just look at their connectivity profiles, the average group connectivity, you see this frontal projection to the STN, but there's really not a clear difference between disorders. But if you now apply fiber filtering and coat the fibers with what is relevant for treatment improvement, you see a clear gradient that you have to stimulate more frontally for OCD and more posterior for dystonia. So M1 or even S1 receiving sites for dystonia, and then, SMA-ish, pre-SMA-ish regions for Parkinson's disease, and OCD is really more DACC or VLPFC and so on. So more frontal, very frontal projections. So this is, of course, interesting neuroscientifically to kind of know which cortical regions play a role in these disorders, but it also is very clinically relevant to know where to put electrodes in these disorders, right? It's always in this nucleus, but we have to fine-tune the target within there as well. And so for the next 5 minutes or so, I want to focus on Parkinson's disease, zoom in on just that disorder, and that study did not introduce this SMA link. It was kind of known before for PD, and we knew it, others knew it, and there's kind of this sweet spot in that region there in the subthalamic nucleus that the world now agrees upon somehow. It's really something we do agree on. This is one paper from us, but there's lots of people that have shown very similar results by now. And if we now project this to this 3-dimensional image of the subthalamic nucleus, and that's the sweet spot, and now you guys have a patient in front of you in your private practice, and they have the electrode placed here, you would be able to tell which contacts to activate, right? It's a trivial problem. We'll take the ones that are close to the sweet spot. So these maybe, or maybe if we look closer, maybe these two, if we turn the electrode around, maybe we think, oh, can actually use these 2 electrodes to steer into this kind of sweet spot, right? So this is a trivial problem. You could all do it without being MDs, so a computer could also do it. So we can build algorithms that choose this for us, right? We can create these models of where the electrodes are and then where the sweet spot is, and then we can let the computer test everything in silico, use gradient descent or other things to find the best contact. And we did so in a first algorithm called StimFit with Jan Rödiger, also here at Charité, and Andrea Kühn, my former mentor, then also did a prospective clinical trial where they actually tested it, right? So they used LeadTBS and StimFit together against the clinician, and both put on settings and showed that both were the same, right? There was no inferiority for the automated parameter suggestion. So this is just to show you that this technology is ripe to actually be employed in patients and helps patients already. But some of you that are more awake might now think, but Parkinson's is different. They have different, there's not one thing, they are not all the same. There's not this one target to rule them all. But there is different symptoms. There's tremor, there's bradykinesia, there's rigidity, and they also have non-motor symptoms. They may have depression, they may have cognitive decline, right? So it's a very, diverse disorder. So there might be something like this, a segregation of this Parkinsonian loop into subcircuits. And indeed, we did look at this. This is already the result of this study, where Naditha Rajamani pooled an even larger cohort of ST-NDBS cases just for Parkinson's, looked at the specific symptoms, and came up with this clear pattern with very interesting details. I don't want to bore you with the details, because I think nobody's really interested in the subthalamic nucleus, clinical applications, but you could really see that specific symptoms like bradykinesia would, let's say, be on one side of the nucleus and a bit more posterior and so on. There's also very interesting connections there, for example, that the tremor tract connects to the cerebellum, as known, as I've shown in the beginning, and the gate fibers would connect to the brainstem PPN region. So it kind of all makes sense. So the way I think of this now clinically is that this is deep brain stimulation right now. One target to rule them all. We have hopefully a well-placed electrode. We put a lot of current on it, stimulate it all, help it all. It kind of works. But in the future, we could be much more deliberate, where we could say, along these new electrodes that just entered the market with 16 contacts, we could deliberately shape the current to different circuits that play a role in the symptoms that the patient has, right? So the patient has a lot of tremor, we will choose the tremor circuit, right? A patient has a lot of bradykinesia, we will choose a different circuit. Could even apply different frequencies on these circuits on the same electrode. That's technically already possible, because we know that some symptoms do respond to different frequencies as well. So this is really not lived yet, right? So what is lived is the left, where we just put it on and fine-tune it a little bit, but there's no deliberate programming. And within that same study, we came up with an algorithm called ClearTune that would test in silico all the solutions we have inside that structure, test the predictions of the circuits that we stimulate, and then essentially come up with the optimal setting. You can see it converges on a specific setting that's also pretty close to the sweet spot. But the cool thing is we can now rerun the same algorithm for the same patient. The electrode is the same thing, but we can tell it now optimize for tremor, or now optimize for bradykinesia, now optimize for rigidity. And each time it will find different symptoms. So even as the disease progresses, many people will start with tremor as the main chief complaint, and then over the years, maybe gait will become their chief complaint. We can rerun ClearTune in the same electrode and find a new setting for a patient over the time as time progresses. All right, we can also zoom into OCD in the same way, right? Before we do so, I want to talk a little bit about that red tract you see there, because that tract even also wasn't really new in that study, right? The other two were new, so there was something new in that study, but the PD loop and the OCD loop had been described before by us. And so the OCD one had a long history in our lab, where in OCD it's kind of the opposite of what we had before. Before we had, you can put electrodes into the same target and help 4 disorders. That's weird, right? In OCD, the story is that you can put electrodes in different parts of the brain and all seem to work. Also sounds like placebo in a way, right? It doesn't seem like there's a surrogate, but if you look more closely, you'll find they all connect to the same network, right? So it's really this idea that I briefly hinted at in the beginning. We can also stimulate at different nodes of the network once we know the network. But here what happened is people did not know the network or just had ideas about it and tested various sites, and some worked, some did not work, and that also could be used to shape out the network, right? So what we did to look at this was a first was my last author paper as a Minota group leader. So we started small. We had 2 sites, the anterior limb of the internal capsule and the subthalamic nucleus from Grenoble. Not huge cohorts as well, but that's, it's OCD DBS, so that's not, done every day and so on in the world. And then over time, we had first results, and we had rotational students. We had, I gave talks about this, who could augment it with 2 additional cohorts. So what we did find here is if you just take the connectivity profile from these electrodes, you get something different. That's not surprising. If 2 different sites in the brain, you seed from them, you get different networks, sure. If you do fiber filtering, though, you see the same red bundle in both of them independently, right? So in 2 different regions, filtering the fibers that would lead to treatment success would give you the same answer. And that was super exciting to us, right? There was a eureka moment. We could actually cross-predict. We could train, calculate the network on the ALIC patients and then predict the outcome of the SDN patients and vice versa. This was the first to use the Connectome to kind of cross-predict clinical improvements across DBS targets. We could also use this kind of combined cohort, predict the other 2 that came later. There's proof because there are preprints without these cohorts, right, with the same, showing already the same tract, and it was also a long history to get this published. So, we had to preprint out much earlier than the first paper. So when the paper came out, within 6 months, there were 4 replications of this by others because people had the preprint and they were kind of preparing to use the tract. We published the tract openly in LeadDBS, and they could download it, test it on their data, and so on. So we were super excited when the first one came out from Mount Sinai, where they essentially downloaded LeadDBS, localized their 10 electrodes, tested how much did these stimulate our tract or not, and that explained the outcomes in their cohort. The same was true in Maastricht, and then in Queensland, even with a different target. These were implanted slightly more ventrally in the bed nucleus of stria terminalis. And then, interestingly, even in patients with Tourette syndrome, implanted to the GPI at a very different place, right, but has fibers passed there in the anterior border. Tourette's patients have obsessive-compulsive behavior as well sometimes, and so it also explained their obsessive-compulsive behavior. We could even double down on that, right? Recalculate the same tract on these Tourette's patients, got the same answer, and then we used the published OCD tract to explain the outcomes in the Tourette's patients, and that also significantly worked. So kind of seems like this is transdiagnostic, right? It's more about the symptom than about the underlying disease, and we see that in many disorders like Parkinson's as well. The tremor tract is really the same in essential tremor and Parkinson's tremor. And then obsessive-compulsive behavior also seems to kind of live there, no matter if you have OCD or Tourette's. Okay, so the last thing we did on this was a blinded replication. I find it quite interesting because this is, you have to think about OCD DBS is something that you cannot do that fast. There are not many patients that want to undergo it. So good centers have like 5 patients total. Great centers have like 10 patients total, and they would do maybe 3 patients a year. So you can think of prospective trials in that domain is really, really tough. What we thought of, what we could do instead of a prospective trial to still make sure this is really true is a blinded replication. So we were in Boston, Samir Sheth was in Texas, Baylor, Texas. He had 10 additional DBS patients that we had never seen. He just sent us the neuroimaging data and the stimulation parameters. We didn't know anything else, no age, no sex, nothing, right? So nothing about these patients. We ran them through this tract model, predicted their outcome, and sent our predictions back to Texas in a very nervous email because he only can send it once, right? He can't say, we want to try again, right? It's one shot. And sure enough, Samir's team knew the actual clinical outcome, and both teams were super excited when our predictions correlated with the real empirical improvements. Just 10 patients, but really done as rigorously as we could. And the next thing that happened was a phone call from Sao Paulo where 2 dear friends and surgeons did implant an electrode using our target and also the results from Volker Kühnen in Freiburg as their kind of guidance, put it in directly in front of the SDN, switched it on, and had a remarkably good response in a month. It was just a 77% improvement. That's a lot. So for people in OCD, even 40% is a lot, right? So 77% is almost gone in a way, right? So this is really remarkable in that score. Patients still doing well, and yeah, this is a great success for us. I was then at MGH at the time in Boston, and they had also implanted patients, so we could look at where were these patients stimulated, right? And this was one patient that was stimulated. The activation, as you can see, the red bubble there, was even in the negative fibers. Also didn't really improve that well, but it's very hard to find the right settings because you don't see an immediate effect normally. So we called her in, stimulated on the different side, maximizing engagement of this tremor circuit, and that even made it to CNN. I think I'll play this video.
A young woman once confined to a wheelchair with a severe neurological disorder is now back on her feet. There she is right there celebrating her GED, and it turns out that researchers were able to use a brand new map of the brain to dramatically improve her treatment.
But then new research of a new map of the brain was published from researchers at Mass General Hospital and around the world. Researchers used this brain, which essentially looked at the nerve networks that seem to be dysregulated or dysfunctioning in 4 different disorders. And they used this map to adjust the deep brain stimulators for 3 different patients, including Julia. After they adjusted her deep brain stimulator, Julia telling our senior writer, Brenda Goodman, quote, "It gave me my hope back." And of course she did.
So she is actually a patient advocate, Julia. So I admire her work in this field, standing up for OCD. And she told us and the team of MGB that something clicked. I could finally focus. Once the DBS was calibrated, I was finally able to really work with all the amazing patient staff that work with me in the hospital. So it opened for her a window to be more receptive to talk therapy, behavioral speech therapy, and really improved her symptoms as well. So after this story, of course, the surgical team decided to use this information also prospectively in surgeries. They have essentially now a routine that would take the tract, burn it into the DICOM image, and then use it for planning there's a prospective trial going on in Brisbane, also with this in mind, one at UPenn by Casey Halpern that has a different focus, but also still uses these tracts as guidance at least. And I personally do collaborate with Ben Shofty, who's a dear colleague in Utah, and he has started both DBS and lesion surgeries, and I essentially help him plan. He's fantastic. I don't think he needs my help, but this is a scientific, very interesting endeavor. I essentially put the electrodes, so the tract in there, give him a coordinate. He would still plan independently, often come to the very same conclusion. But so far, the 5 patients we've done together, or he's operated on, were really all successful. Here's one email, just showing how excited he was about one of the first cases. So this really seems to work, right? Hopefully by now you kind of believe me. It's all retrospective evidence. It's all low case numbers, I know. But coming short of a prospective trial, this is still very, very promising. But now that maybe you believe this tract works for OCD, I want to tear it down again and say it's all, just a first step. And same applies then in Parkinson's. Every patient is different, right? So some have more obsessions, some have more compulsions and depression and anxiety. So while this tract might work for the average OCD patient, on average, it might not be a personalized target for every single individual patient. So similar as in Parkinson's, we essentially went all in, got the largest cohort together from dear colleagues around the world. These are just the electrodes. These are just the active contacts. You can see this is a lot for DBS, so lots of stimulation sites. And we essentially came up with this symptom-specific model where you could see that symptoms fan out, again, as a gradient within that internal capsule, so anterior limb of the internal capsule. Some being more receptive of treating obsessions, some more for compulsions, depression, and anxiety. There's even 2 more symptoms, cognitive flexibility and control, that we don't trust that much because we didn't have enough data. But the idea is that all of these kind of tie into this global functioning of the patient, but we could fine tailor to the individual where to stimulate, which tract to stimulate at which site. And this is the same idea, right? We have a patient coming into your office. They have a lot of obsessions here, maybe also a lot of anxiety, as in the first example. You would take these two networks, weight them higher than the other two. You would find the optimal spot where they converge and switch on these electrodes, right? Same if, things are different in the second patient. And we can use ClearTune, the same thing as we've done in Parkinson's as well, to find the optimal treatment for an individual patient. Of course, we can also blow this up in the future to have even more symptoms, more fine-grained tract information, but by doing so, personalize treatment. So going from this one-size-fits-all tract to this kind of gradient tract. Yeah, I won't zoom into dystonia long. I'm sorry, I won't bore you with everything. So I think this was a lot already. So just to briefly mention, we also did similar things in dystonia. That's all I want to say here. I won't go into details. Some very interesting things about even Nico Dosenbach's CAN network and the M1 phylogeny. So very, very interesting stuff, but I won't bore you with it today. But just to show this vision now is that we can map what we call the human dysfunctome, dysfunctional circuits of the human brain across disorders using techniques such as DBS. And we need your help, and we need the help of the world. We can't do this as a lab alone. But essentially, the hope is to create an exhaustive atlas of symptom-specific circuits that play a role that we need to downregulate when they become dysfunctional. All right, I have to speed up a bit, but yeah, it won't be much longer, but I think this might be interesting to you too. So can stroke lesions also inform DBS? I did show you this in the beginning, this brain lesions here, right? Causal evidence. We have a stroke. We get, let's say, aphasia, and we have that again in the second patient and again. You could say, okay, we take these lesions, overlap them, and that's where aphasia lives, right? That has been done for centuries. And it still works, but often the result is like this, where lesions just don't overlap. The lesions that produce the same symptom could be anywhere in the brain. Mike Fox, my dear mentor in Boston, came up with this idea of lesion network mapping and asked instead, would they all map to the same network, right? Can we find one network that kind of hones down to all these lesions? And that is often the case, right? So these are rare stroke events. I even had one patient myself here in Berlin that had a small stroke in the thalamus, and all that happened was that the tremor stopped. So it was even a good stroke, you could say, although no stroke is good, but that patient had tremor before and then had a small stroke in the thalamus, and then tremor was gone next day. Very interesting. These are all cases like this from the literature. So these cases could tell us something very important for treatment because we could mimic that stroke to treat tremor. And when we look at their networks, the peak sites where they all converge, where they're all connected to, are in the thalamus, in this small region, in the VIM, and then in the cerebellum as well. And that's precisely where we put electrodes, in the thalamus, and people are investigating the cerebellum as well. This is the lesion network map, just thresholded increasingly, and you can see throughout the entire brain there's really this one pixel surviving, and that's where people have put electrodes for decades now, right? So this seems to be a recipe to come from strokes to treatment sites. And we've shown it for tremor where we already knew where to stimulate, but we could do this for other things as well. And Mike has shown, for example, lesions that cause cessation of addiction, right? Where people stopped smoking after a stroke. That could be very important to know where to treat for addiction. Now, most of the strokes would produce a symptom rather than, get rid of a symptom. And these are strokes that led to tics, secondary tics. Christos Ganos is one of the world tic experts, and he has spearheaded this work. And he has curated all of these case reports. They are all literature reports. Looked at all the connectivity from there. Bassam Al-Fatli as well, who might be here, did actually the analysis and was, very involved in the study too. And essentially came up, Bassam and Christos came up with this network of these, sites where all of the lesions that produce tics would be connected to. And as aficionados of DBS, which you are probably not, you could already see this is the 2 sites where we do put electrodes to treat ticks. So we reached out to dear friends in Cologne. I wasn't in Cologne back then, so this was a collaboration with Fearless team and Carlos Balderman, and also other people in Maastricht and Paris, and looked at electrodes. Here you can see the, in petrol, the stimulation side, the VTA, of an electrode, and the lesion network side in yellow. Right? So in the bottom, they missed that network. In the top, they hit the network. And that degree of overlap could explain clinical improvements in both of the targets, so GPI and thalamus. So again, some way of bringing evidence from lesions to therapeutic sites, which I find very, very interesting. But let's finally get to the end of the talk that I promised, right? So I started with the end. You remember this slide, maybe. Great work from Jülich, right? BigBrain and the PLI images from Markus Axer, again in vervet monkeys. It's a bit inflated. I think in human it wouldn't be as bad as shown here, but it would still be bad. How can we mitigate this, right? How can we get to better anatomical models? And I think, you know, the stuff I showed you in the main part of the talk is that we need these, right? We're already kind of successful, but we need to get better. We need to better understand where the electrodes are, which circuits we want to modulate, and also we want to make sure that every single patient succeeds and has great success, and not just our champions like Julia Hum that I showed you. So this is really very important. And one option to do this that the world is trying is to put a lot of money into building better diffusion MRI machines and better algorithms, and maybe even map microscopic things. And I think that's a very valuable endeavor. I don't want to downplay it at all. I was involved in a great, just briefly involved in a very great effort led by Anastasia Yendiki and Susan Haber, where they try exactly that, right? They even used, let's say, our Nature neuroscience study to motivate it, right? To come up with, we need better circuits for these things. That's one way to do it. And while we were part of this project, we had access to their best diffusion scans. And with that, Konstantin Butenko could look at these COM fibers that come from the pallidome and connect to the STN. And for the people that don't know the COM fibers, they are very important, very hard to reconstruct because of diffusion MRI, because the internal capsule comes there, and the internal capsule is a highway. It has a strong diffusion signal, and destroying almost anything that you can see that would go orthogonally to it, right? So they're a good use case. So this was a post-mortem brain scanned over a weekend on the Connectome 2.0 scanner, which is really new. It was just brand new installed there. It's crazy, field strengths, and sorry, not field strengths, gradient strengths, and also scanned by, I would think, one of the best teams in the world at Martinos, right? So it's really as good as it gets. And they look great. We were super excited to see them, but the reality is that they look like this. They're much more fine-grained. The video is not playing well, but you can see like more hairy fibers, very thin fibers going through there, that I think have a functional importance. Maybe they're not as random as they look like. Maybe some of them are the ones that we need to simulate for Parkinson's, others for OCD, right? So they might really play a role, but we don't see them. And even with this great diffusion data, it just doesn't look like histology like this, right? It's beautiful normally. It looks like an eye. And then, even if we could do it with post-mortem MRI, we won't be able to get there anytime soon with patients, right? So one point I want to make is that atlases will always be superior to individual scans, because if individual scans get better, atlases will also get better, right? And we have more time. We can scan over weekends in atlases. We can have a dead brain that lies perfectly still in the scanner, right? So this is my brain, again, scanned by maybe the best team, John Polimeni, who, you might know is really even cardiac-gated flash, specialized, really beautiful. You can see these fibers there really well. But this is a dead brain scanned by the same team over 100 hours in the scanner, and this is much more fine-grained, much more beautiful, right? So atlases will always be better. And so the alternative solution And this is not just atlases from MRI. I think histology will always be better than MRI too, right? So the alternative solution would be to say we know the anatomy, we take it into patients and build very precise atlases without even using diffusion MRI in the case of fiber tracts. And this was pioneered by the team of Cameron McIntyre and Mikkel Petersen first authored the report. I think it's been a paper that's, for me, been very, very important, but maybe hasn't received the attention it would deserve. They would essentially take primate tracing data from Susan Haber and recreate 3D models out of thin air, so without diffusion MRI, in the computer by creating these streamlines that you can see here in the top and then making them fuzzy. And then they discussed with people like Susan Haber, with holographic lenses, where the fine details should go. Right? So it's really creating fantastic atlases. So as a reminder, none of these tracts were actually made based on diffusion MRI. And so what I think we should do is exactly that. I think we need a different name for this than tractography, because if not, people confuse it. Cameron tells me that people tell him all the time that his paper is great tractography results, but it's not diffusion-based at all. Maybe a term like architectonics could be good, because we're really more like architects. We build the tracts in like resources as the big brain that you're building. We should ground them based on anatomical data, and we could then apply them to DBS. So these are just two examples where we did this that I won't go into detail, but we had the evidence from anatomy. We came up with hypotheses based on it, and then looked at which of the hypotheses came true. So when Cameron McIntyre published that paper, I was struck and I thought, okay, we have to probably kick diffusion MRI out of the window almost entirely, and this is the way. And we also as a lab launched a big effort that led into this focus atlas where we created very meticulous definitions of gray matter structures in the MNI brain. And we used various resources, including the great resources you guys are creating. We focused on these clinical intervention zones where we know we will put electrodes there, right? So we can't map everything in precise detail, but we can focus on these intervention zones. And then we added these manually traced tracts. So these are really streamlines that we put in 3D Slicer, and then we made them fuzzy with the software we created that's openly available, Curve to Bundle. And we used, we really went for these very thin bundles to the histology, to post-mortem scans and so on, to make sure that these are more precise as possible, leading to these better anatomical definitions of white matter. And this is kind of the total atlas, as you can see it here. Very, delicate details of, also the advantages that the fibers can go through the gray matter without a problem. There's no loss of diffusion signal there, and so on. So lots of really detailed fibers, and none of them have been done with diffusion MRI. And I've kind of done this out of lost hope, right? I'm not an anatomy lab. We're not an anatomy lab. I'm not an anatomist. There's just nobody doing this, and I think you guys are doing it, so you should. We need this. We need to refine this. We need to make it better. We need to make sure it's better, it's correct. And we should also come up with even better atlases that you have the resources to produce, right? So we need these fiber atlases that are in MNI space. And if people tell me, well, but we have to be patient-specific, then I would just show them this picture and say, well, try it with your diffusion data from a patient that moves in the clinical 1.5 Tesla scanner. It just doesn't work, right? And I think this is maybe just the beginning, streamlines, and what we should actually have, this is a slide from a talk by Max Schein that I just had a picture taken. I think we should actually even create models of cells where we have the synapses, where we have their transmitters and their axon thicknesses and so on. And then we can model really what happens there with electricity being applied. And they will never be patient-specific. It's not possible. But we will never get to patient-specific level of that, or never say never, but not soon. So to conclude, we really need you. Feel motivated to create normative atlases. They are needed in surgery, even if maybe my dear colleagues sometimes don't know it in the field. And I hope I could convince you that using this deep brain stimulation network mapping, we can study the organization of the human brain and the dysfunctome. We can map symptoms across diseases, and we need better anatomical models. And with that, I want to thank my group. This is still my Boston group, but they did most of the work that I showed today. And the new logo in Cologne, feel free to visit, come from Jülich especially, but also from the world, and happy to take any questions. Thanks again for the invitation.